Thank you very much, Covaris, for inviting me to come speak to you today. And I'm going to talk to you about a scalable FFP sample prep method that we are implementing into our clinical LCMS assays. So I'm coming from Memorial Sloan Kettering, and so we're a cancer treatment and research institute, and we're the largest and oldest private cancer center in the world. And we're comprised of Memorial Hospital, which was originally New York Cancer Hospital, and Sloan Kettering Institute. And those two institutions merged in 1980. And so I'm a member of the actual hospital, and I am the director of the clinical proteomics laboratory. So our laboratory is part of the hematopathology service within the Department of Pathology and Laboratory Medicine. And until a few months ago, we were just the Department of Pathology, but we have merged. So I look at this within a perspective of pathology and pathological diagnosis. And we are regulated by New York State Department of Health. We are a CLIA lab, so we are very highly regulated, have to follow a lot of standards. And all of our technologists have to be licensed by New York State, which means a lot of them are coming from a clinical laboratory training background, but do not have mass spectrometry experience and not proteomics experience. So that's why I think this will show you that this type of workflow will work really well in an environment where you're not necessarily specialized either. So here I just show you our Q-Exactive, our laser microdissection, LMD 6500. I won't talk about microdissected specimen today, but we utilize microdissection in our assays. And then we have an LE220 RSC from Covaris. And all of the work I'm showing here has been done on this system. So when you think about pathology and you think about what pathologists are doing and how diagnosis is being formed, you probably primarily think about microscopy. But in reality, modern pathology departments and diagnosis that is occurring in these departments is more complicated. Pathologists are looking at microscopy, they're looking at morphology, they're identifying cell types, things like that. But there's also cytogenetics, especially in a cancer hospital. We're looking at chromosomal abnormalities, flow cytometry to identify unique cell populations and abnormal cell populations in molecular pathology. So we're talking about next generation sequencing, looking for mutations that help further refine the diagnosis or subtype of disease, especially in things like lymphoma and leukemia. And then this is just an example here, sorry, there's a laser, of clonality testing on lymphomas. So what we are trying to do is look at how pathology already incorporates these advanced technologies in diagnosis, and World Health Organization and other bodies already require some of these advanced diagnosis, things such as molecular pathology for gold standard diagnosis. And what we want to do is take mass spectrometry-based proteomics and move it into that world. And so utilizing patient specimen, that standard patient specimen, FFP tissue primarily, fresh frozen occasionally, serum, blood, and then cells coming from bone marrow biopsy, CSF, et cetera. And what we are working really focused on is pre-analytical processing of these samples so that they're amenable for LCMS analysis. And then, of course, the development of the actual assays using the instrumentation and how to generate that data that is going to mean something to our pathologists and clinician colleagues. So today, I really am going to just talk about how we're utilizing the Covaris system for processing and preparation of our FFP tissue samples. So if you've worked with FFP tissue or you want to work with FFP tissue, you're probably quite aware of the fact that the tissue goes through a whole lot of processing, and we have to think about all of that processing when we want to go backwards and extract out the protein. So one of the benefits of being part of a pathology department is that I have been allowed to go into the labs and see all of the different parts of the processing, but just briefly, tissue specimens get grossed, they get fixed, dehydrated, embedded, and then cutting, staining, and then review. So we kind of take the sample somewhere around here and process them from there. So about 5 years ago, we did our first validation on a clinical assay, and that's from FFPE tissue. And this is the protocol that we have been following since then on this sample prep, and that we optimize at that time. So we start with a 2-step heating for reversing of cross-linking. All of this is done in 0.5 mil tubes. We use a bath sonicator, and so tubes are floating around in there. We do reduction alkylation, and then an overnight trypsin digestion in C18. So we're doing all this on a thermomixer in tubes, so we're limited to about 24 samples per thermomixer. Total time, about 16 to 24 hours, because depending upon how early we are in and moving forward with C18 cleanup. So when you think about a clinical timeline and turnaround time, this is not really gonna work because pathologists and clinicians are waiting for answers. And if if an immunohistochemistry is gonna come out the next morning and we're taking 3 or more days, that's not gonna be very helpful. So what we wanted to do is take this sample prep and put it onto our Covaris LE220 system. So the first step of that process is to take and transfer this homogenization step onto into the AFA. So we we have the same heating step, but instead of doing 30 minutes in the bath sonicator, we have 2 steps of AFA. And so this is using a TPX 96 well plate. And this is, so if we do 1 row, it's 5.5 minutes of sonication on each side. And a whole plate you can do in 66 minutes. So it's 96 samples. So we did direct comparison of just the homogenization where we left everything else the same. And if we look at total protein extraction, so this is using a healthy spleen sample, and we just take a 6 micron section. And we look at total protein extracted based on a BCA assay. And this is we use a zwitterionic buffer. This is what we originally validated. So we just wanted to do a direct transfer, keeping buffer system the same. And then if we look at, we normalize and run the same amount of the sample by LCMS on our Q-exactive. And we see that protein groups and peptide groups, unique protein and peptide groups identified in these replicates are pretty similar, maybe slightly lower when you look at the AFA. But we did then look at what proteins are identified in at least 3 out of 4 replicates of any condition. And when you look at that and take out that stochastic effect, that then you have pretty high overlap. So basically what I'm saying is that we're extracting the same amount of protein using either method, and then our protein profile that we get from this sample that we've extracted is very much the same. And we also specifically look at certain protein groups that we're more interested in. And so one group of proteins that we pulled out and looked at, log2 abundance is what I'm showing here. And this is just from within Proteome Discover doing label-free quantitation, pretty uncomplicated analysis. And we looked at immunoglobulin subtypes. So these are constant regions. So it's pretty similar between the 2 methods there. So we did some initial, I'm not going to bore you with all of that, but we did some initial optimization of our specific homogenization to further improve the protein extraction. And once we were happy with that, then after some additional optimization of shortening our heating and shortening our reduction and alkylation time, we moved on to what is our real bottleneck, and that is our digestion. So doing overnight trypsin digestion is going to obviously not allow you to get your samples onto the instrument in the same day. So we wanted to move that step onto the Covaris with the AFA and do trypsin digest with AFA. And so in this case, we again are using a healthy spleen sample, same buffer system. In this case, all samples are in the TPX plate because they're all going on to, the whole workflow is in that 96 well plate format. So we compared overnight digestion at 37 degrees And we compare, and then 3 hours at 37 degrees on the thermomixer, and an hour of AFA digestion in 3 hours. And so we looked at, focused on, I'm not showing, but protein group identification, all of that, when we analyzed it by mass spectrometry, was very similar. And so we focused in on miscleavage. And so we see that with the overnight digestion, the lowest amount of miscleavage, but 3 hour digestion on the AFA is maybe a couple percent more miscleavage. So we're talking about less than 10% miscleavage when we do digestion with 3 hours of AFA. And then we spike into our samples a digestion standard from Promise Proteomics called Digestive. It's heavy isotope labeled. And so we have this tail here where it has varying degrees of difficulty triptych cleavage sites. And this is just showing older data when we first started using this, and we did some time points. So I'm just showing some of the time points from that, and where we tried to see what miscleavages are we most likely to see if we have any digestion issues. This last pair here of the miscleaved peptide and the fold cleavage are the ones, but we target the set in all of our samples to evaluate any kind of trypsin digestion issues, and that's true of all clinical samples. So if we catch any issues before analysis or during analysis. So this is just showing the percent of that peptide that is miscleavage product versus fully cleaved. And so in the overnight and the 3 hour AFA, we don't detect, or any miscleavage product of that pair is below our limit of detection. So we feel like our digestion efficiency is comparable between overnight at 37 degrees and 3 hours on AFA. So what we were able to do is take a workflow that we were having run overnight and now fit that into like a 7 to 8 hour workflow and able to go from something that was limited to 24 samples roughly. We could do multiple thermomixers, but then you're also dealing with like 48 tubes you're opening and closing all the time. Now we do this on a TPX plate all the way through. We put the samples in here, and they come out once we get to C18 cleanup. So now we can scale up to 96 samples. And so the AFA has facilitated a workflow that's allowing us, to do homogenization through digestion and able to do this in a single well, so we're not transferring the samples at any point. And then also this would be compatible with automation. We don't have automation at this time, but that is best to kind of develop these methods so we can move it to that. And so we have this high throughput workflow. And we can use this workflow. Some tests we have are low volume. And then we have other tests that we're in the process of approval or validation that might be very high volume. So we can use the same workflow, the same processing for a wide variety of sample numbers. So I'm just going to show you how we've implementing this into an assay that we've already validated and we're working through getting approval for. So plasma cell neoplasms are diseases which abnormal plasma cells form tumors in the bones. And multiple myeloma is the most common form of plasma cell neoplasm. And plasma cells, if you don't know, are your white cells that are producing antibodies to fight against infection. And the neoplastic plasma cells accumulate in their bone marrow and they displace your healthy cells. And that neoplastic clone just keeps replicating. It's producing a monoclonal antibody. And most are producing, and they're producing a monoclonal antibody, and most are secreting high levels of that monoclonal antibody. And so diagnosis of a plasma cell neoplasm requires the identification of a neoplastic monoclonal cell population by microscopic evaluation of the biopsy, and then characterization of that monoclonal immunoglobulin, usually by immunohistochemistry, so antibody-based detection on the biopsy is shown here. Or in situ hybridization. And so the immunohistochemistry assays for immunoglobulins tend to have very high background staining because they have some issues with specificity. They're using polyclonal antibodies in many cases. And also these assays aren't standardized and they're very difficult to make quantitative. So what we wanted to do is to develop a targeted mass spectrometry assay that allow us to determine from the tissue the relative quantitation of immunoglobulin heavy and light chains so that we can characterize the immunoglobulin in the tissue, especially when some of the immunohistochemistry is difficult to read. So we just developed a parallel reaction monitoring assay where we looked at the theoretical digest of those proteins, right? We looked at what proteins were unique. We used purified immunoglobulins of different subtypes to do test experiments and finalized our list of peptides and ordered stable isotope labeled peptides. And then we did all of our LOD, LOQ, reproducibility studies, et cetera, and ended up with our table here showing all of the peptides that are part of the targeted assay, as well as their limit of detection and limit of quantitation in femtomoles. And so we process our FFPE bone marrow needle biopsy, which is quite a small biopsy, and we take one section at 6 microns of that and process it. We spike in these heavy isotope labeled peptides, run that in a scheduled PRM method on the Q-exactive, and then we analyze in Skyline. And so we look at relative abundance of these different heavy chain subtypes, the light chain, and then also immunoglobulin gamma subtypes. Not relevant to so much to plasma cell neoplasms, but some other diseases that we're also validating for. So when we did this validation, we're comparing the results to immunohistochemistry results, because these are all cases that have been diagnosed and results signed out. So for example, in this case, we see that it's the light chain is lambda, which matches with what the immunohistochemistry is. So after we have our results, we compare that. And then also we compare to serum studies, which are always done on multiple myeloma patients, where they look to identify if there's a monoclonal immunoglobulin circulating. They determine the subtype by immunofixation, which determines that's IgG lambda. And then we look at free light chain levels. These are all used in the diagnosis and characterization of that monoclonal antibody. And we compare that to what we are able to do by mass spectrometry, because these should be the same monoclonal antibody. And so we can see that those align, the serum studies align with what we are seeing from our tissue analysis. So we had a validation set of 44 FFP samples. This work was done prior to us fully optimizing our FFP method on our LE220 RSC, but we took a subset of those samples and processed them following this workflow. And so this is hard to see, but we'll show one. So we did 15 cases plus a myometrium negative control. And all the results were concordant, meaning that what we would call that immunoglobulin subtypes matches with between the two sample preps, and that the results were the same regardless of which way we processed our tissue. So this is just one example of a case where there was a woman who came in with spinal cord compression with concern for multiple myeloma. Bone marrow biopsy shows a high involvement of CD1 of neoplastic plasma cell population, and it's lambda light chain restricted. And we see that on the left is when we did our initial validation, and on the right is the relative percentage of the subtypes based on the sample prepped using our improved workflow. So the results are concordant regardless of which way we process the sample. So we did all of our validation prepping our sample one way, and then we switched it to our high throughput method, which will really increase the throughput possibility of this assay, which is expected to be quite high, decreases the turnaround time, making it possible for FFP-based proteomics to fit within a true clinical setting and expectation. I just want to show that we also utilize the LE220 RSC for a lot of different sample prep types. And so we have moved, there we go. We've developed our cell lysis workflow as well on the system using the TPX 96 well plate, including, this is an SDS-based buffer for this example, so we are using SP3 cleanup and doing our digestion on bead in the system as well. I'm not going to show any data about that, but I just wanted to mention that because we are trying to use this for a lot of different sample preps. And so this is allowing us to do diverse sample prep in short turnaround time with high throughput. So I want to just thank my colleagues in hematopathology at MSK, Cooper, technologist in the lab, him and I did most of these experiments together. The surgical pathology laboratory, which allows me, like I said, to come in and ask all kinds of weird random questions and to help me get materials as needed. And then my colleagues at Covaris who worked with me on this, and of course the Farmer Family Foundation, who funds our protein diagnostic development. And there's a poster with a lot more details and data about the FFPE development steps later today. And if anyone has any questions.